we'll start. So thank you for coming. It's my pleasure to introduce you to a group of, it's called CDG alias Pain. It's a network which is established here. And today we have Professor Jack Jacken that came to visit us. And we also have Vanessa Ferreira that we are co-directing this network. And there will be other introductions, but I have to say that I'm really honored for this moment, and we also have other participants from this network here together. And thanks to Professor Jack Jacken for all the support. And this is again another support to our efforts and this seminar, and the fact that he's with us, listening to us, and also involved with the researchers, with the families, and so this is a great honor for us.
Okay, so I mean, for us, for Paula is a great honor. For us, it's a super great honor, because we are since Saturday we are really close in a room talking only about CDG research. So it's, we are celebrating the first CDG lab retreat from our research network. And this was a huge commitment because you will see, I will present later, but we have our postdoc from Italy coming, our postdoc from Azores coming. So it was really like everybody really wanted to be here with you and to present their work and to brainstorm about the research that we are leading, which is innovative research. So just for letting you a little bit hear more about us, because I'm not sure that everybody is in the same page, but actually all this story happened because of unconditional love. So in this case is unconditional love that I have for my sister who suffers CDG, which stands for Congenital Disorders of Glycosylation. And I brought this sentence because last February I was in Las Vegas, in the old town part of Las Vegas, and there was a guy saying, "Let love rule." You know, and this is the kind of situation you walk around and you say, "Oh, this is familiar for me." I mean, let love rule, but why? And then I went to search on internet, and it ends up that it is a music from Lenny Kravitz that more or less says, "Love is gentle as a rose, and love can conquer any war. And love can make a little child," in this case also CDG adults, smile, "but we got to be strong, and we can't do it alone." And this is exactly what happens with our CDG International Research Network. We cannot do it alone. So as I said, everything started because my sister, she's now 37 years old. She will turn on end January 38 years old. And her birthdays are always a big achievement for us as a family, because we were told 20 years ago that she would not live more than 18 years old. So for us, it is really an achievement. So because we are a very small group, I wanted to share with you what happened. So obviously, at the beginning, this is what my family planned for me. So basically, you have your parents, they have a lot of dreams for you. In my case, I was supposed to be a dentist and many other things. And obviously, I did the opposite. So I didn't study to be dentist, I studied to be biologist. I did my PhD in cell biology because I really wanted to learn more about glycosylation. What is this post-translational modification that happens in proteins? And then later on, I managed to complement the science with an MBA, because if you want to be professional as a non-profit organization, you have to have business skills. Otherwise, it will not work in this competitive world, because it is really competitive. So, and I wondered this morning when I was preparing the slides, I wanted to ask you why you are actually here, because I suppose you have many other stuff to do. So why did you come to this call of action, let's say? Don't be shy. Angelina, queres dizer alguma coisa? No. Because we really believe that by accelerating research, we can do a difference in the CDG and adults' lives. This is why we are here. And one of the reasons is this cutie girl called Remy from Dubai, who is now 4 years old, and she is a PMM2 CDG patient. And now the question is why we all together are here. Because we envision a present and a future where all patients and their family members, they have rights to live a happy and fulfilled life. We want to keep our kids and their parents happy with a meaningful life like the other families. What is our wildest dream? What do you think we want? Yes, we say therapies because I don't know yet a disease with a cure, and yeah, sometimes it's misunderstood by the families. So, therapies. We want fast access to therapies. We want the CDG patients to have the same opportunities than any other citizen in our society. As you probably perceive, CDG is a very severe disease, and inclusion, we are not yet in an inclusive society. So we face many, many difficulties in parallel with a rare disease diagnosis. So, and to have fast therapies, we need to be all on board. The patient, our researchers, that you know some of them, and the medical community, Professor Yaken, and also the other collaborators in our network. So we have set up the patient group in Portugal in 2010 with one single goal, which was to boost awareness with the community united to lead resources and research in order to educate and empower the community. And all the time when we say about community is the family and the medical community, because many of our medical doctors, they use our documents for themselves and also to educate the families. And I wanted to share why we exist as a network, because at the end of 2015, at some point after leading a lot of activities under the scope of the Portuguese Association, I started to really brainstorm about the CDG therapeutic research challenges. And I ended up with a list of 10 main challenges. And when you are a family member desperate to have a good quality of life, I mean, you find yourself somehow in the borderline of a building when you see that we don't still have yet an animal model for CDG that really mimics the most common CDG type and many other challenges that we really need to overcome together in order to make it happen. And how we make it happen? In a united way, with all stakeholders united. So in 2016, and I'm about to finish, I started to talk with Paula and say to her, you know, we are really doing a lot of educational and awareness, but now we have to move forward and really do research. Paula was one of my little angels that supported immediately the idea, but obviously you always have people really not, you know, like, I mean, you are hallucinating, this is not, I mean, yeah, okay, you will do it, let's see, and so on. But then, not only Paula was on board, but also we have Dulce that is here with us, Esmeralda Martins that also was very supportive since the beginning. So at some point you have many little devils, but then you have also the little angels that supports you. And this is where we are. Paula was one of these crazy people that followed my dream with me to make it happen. Also families, I have always to be grateful to this family who managed to fundraise the first 12,000 euros that helped us to generate the Liliana's scientific scholarships. So we always need people that have more or less the same dreams than you, desire and ambition to lift you higher. And then you also have to have pioneer institutions, like your institution here, because you know, a patient group coming to a campus, an university campus, saying that, oh, we want to do research is like completely out of the scope, right? It's really uncommon. So the Nova ID and the full campus, they were supportive, and we have been doing a lot of paperwork to do protocols and to be legal, recognized. We can receive donations directly to a center of cost that is strictly dedicated for CDG research. Is not lost in the big pocket or in the big center of cost. Is everything is for CDG research. And it's my pleasure to present you the group that we have right now. I will ask you to the team to stand up. So we have Eleonora, which is our new collaborator, postdoc. We have Carlota Pascual, which is our PhD student. She won this year an FCT PhD for 4 years, dedicated to CDG. Dorinda, she's now on the way to Algarve, but she's also our postdoc, in this case as a volunteer. And then we have Sandra. So Sandra, she's our postdoc working from Açores. And we have our Rita, our PhD student, that you all know because she's fully dedicated as well, like the others. So to finish, I want to share what I think that we all have together, which is that we share the same values. And the same values is that we have a team that are extremely focused, that want to help each other, and also the CDG community, that run away from the comfort zone to find easy solutions for CDG families, cooperation, curiosity. This was Jack who identified this value, because I completely forgot. So you are completely right, Jack. Altruism and laugh, so to have fun with the work that you are doing, and the sense of urgency, because we really have patients dying. Every month we have CDG children and adults that really die. It's very serious, so we have to put our efforts on it. And Jack, I don't want to put you on the spotlight, but it seems now is your turn to to present.
Okay, thank you, Vanessa, and thank you, Paula. Dear CDG friends and CDG fans, I am going to tell you a story about sugar trees. Sugar trees are very old, not like our ordinary trees, apple trees and so on. These trees are as old as life on Earth. They were already present in unicellular organisms. And this is the simplest possible sugar tree, just a trunk and 2 branches. And you recognize the known monosaccharides, the mannose, the glucose, the galactose, the sialic acid. Sialic acid usually at the end of the branches, and has special properties, namely it has a negative charge. And that is very important for the rest of my talk. Don't be afraid of the next slide, because nature has made it very complicated to make such a sugar tree. Here is our sugar tree, but we start there. In the cytoplasm, here we have the endoplasmic reticulum, and here we have the Golgi apparatus. So it starts there with the mannose pathway, building up first at the outside of the membrane of the ER, then at the inside of the membrane here, intermediate, that goes to the Golgi to be modified. A number of sugars are removed and others are added. Why? nature does this is not really clear, but anyhow, it is the truth. And here is our sugar tree. So what are these glycans doing? You know that our body is full of proteins and fulfilling myriads of functions. About half of these proteins need glycans to be attached and to be present on them to fulfill their function. And you can compare it, in fact, with a train, where the protein would be the compartment with the passengers, and where the glycan is the locomotive. And of course, the most important part is the train, the compartment with the passengers. But when there's no locomotive, this passenger will never arrive. So this is the glycoprotein. Of course, there are also glycolipids, but I will not go into details of the glycolipids. So CDG simply are defects in the glycan formation or the attachment. Here we have the protein without any glycan. And here we have a protein with a defective glycan. So in this case, these proteins cannot function properly, and there is a problem. Glycoproteins, as you know, are present in all organs, in all tissues, particularly in the brain. The brain is really a big, big reservoir of glycoproteins. Does CDG affect all organs and tissues, but unfortunately, particularly the brain. So CDG mostly are brain diseases. So this was an introduction to the main ingredients of my talk. First of all, how did we come to CDG? And as important, what did CDG learn us? So I will try to go a little bit in the early history. Some 40 years ago now, I was asked to investigate these twin sisters because of a problem of psychomotor disability. They were 1.5 years old, and as you can see, lovely children without important dysmorphism, in fact. And I started to do the usual metabolic screening. And on this metabolic screening, I found 2 abnormalities that were very small, but that were, I could reproduce them and not due to medication, namely an increase of a lysosomal enzyme, aryl sulfatase A, and a decrease of TBG, a thyroxine binding globulin. And I could not see any link between these abnormalities. And then I further studied these girls and find other abnormalities also in the sexual hormones. Prolactin, for example, they had a very high prolactin. I mentioned this TBG deficiency. So, in fact, I could not link all these things together, and we published this with a very big title, a long title, as you can see, in 1980, with a suspicion that this could be a new syndrome. Now, on looking into the literature, I came across an interesting paper, namely a paper of Henk van Eyck, a hematologist from Rotterdam, who had published a paper on transferrin isoelectric focusing. Transferrin is a protein that is abundant in the blood and that carries 2 simple glycans with, of course, sialic acid at the end. And he performed an isoelectric focusing. That means that he subjected the transferrin to an electric field. And when you do this, you see mainly tetracyanotransferin in normal people, besides a few others, but the main band is tetracyanotransferin because you have these 4 sialic acid with a negative charge. I thought that this could be interesting for my twins, and I sent serum to Van Eyck, and a few days later he called me with great enthusiasm saying that he had never seen such a pattern. Namely, there was an increase of dicyanotransferin and also of acyanotransferin, pointing to the possibility that there could be on part of the transferrin only one glycan or even no glycans at all. And that was, of course, something very new, and this was the first indication that there could be a problem with glycosylation. And we published this in 1984, and this is still the method of choice for the diagnosis of CDG, and this can help to diagnose CDG in about 60% of the known CDGs. At the same time, I also came into the literature, found Jelena Stibler. Jelena Stibler, at that time, she was a she was a neurologist very interested in chronic alcoholism. And she found out that in chronic alcoholism, you have similar abnormalities in transferrin than in my siblings. So she became very interested in this syndrome, and she looked around in Sweden, and she found a number of patients also in Sweden with the same syndrome. And we published a monograph on this, what we called at that time carbohydrate deficient glycoprotein syndrome, of CDGS, on 27 Belgian and Scandinavian patients. And the main characteristics, you know, of course, are the special morphology in most of them with the inverted nipples, the fat pads. Most of them are also more or less severely intellectually handicapped. They can have hypogonadism, but some of them are also, I would say, have no dysmorphism at all and are looking very well. For example, this Belgian girl could walk alone at the age of 3 years, while most patients never arrive at walking at all. Then, sometime later, I came across this boy, a Belgian boy at the age of 10, with very severe problems. He could not walk, he could not speak, he had a very resistant epilepsy, and he also some stomatological abnormalities. On occasion of a tooth bleeding, the coagulation was studied, and they found abnormalities that were very similar to what we have seen in our siblings. And therefore, of course, transferrin isoelectric focusing was performed. And this was abnormal, but different from our siblings, because there was also an increase of tricyanotransferin and of monocyanotransferin. And therefore, we call this pattern the CDG2 versus CDG1 in the siblings. We published this at that time, but without knowing the defect. And on further study, we found out that this patient had a defect in the making of the second branch. So he could make the first branch, but not the second branch, where you need GLUC-NEC transferase 2. So my hypothesis was a defect in this GLUC-NEC transferase, and I contacted this very charming glycobiologist, Harry Schachter from Toronto, who immediately started and could confirm this in a very short time. And this, in fact, was the first genetic defect in human end glycosylation. But this remains a very rare disease, since we know about, I think, 7 patients, while the other disease, the CDG1, we know more than 900, actually. Still, we had no problem, we had no idea what the problem could be in the siblings. And therefore, I asked Emil van Schaftingen, really a genial biochemist, to look into the first steps of the mannose pathway. I didn't hear anything for about, I think, 8 months. And then he came up with a really absolutely fantastic finding, namely, he found a defect in the second step of the mannose pathway in a phosphomonomutase. And this enzyme was not known until then. So there was a completely, not only a new defect, but also a new enzyme. So this was really an amazing breakthrough. And we reported this in 95. And shortly after, in 97, our, I would say, our brilliant geneticist, Gert Matthijs, was able to clone the gene and to find pathogenic variants in these patients with PMM2 CDG, what we now call PMM2 CDG. So I would say the circle was round, and we had to start another circle, namely finding a treatment. And this is, of course, another story. But in 96, the CDG world was very surprised by this paper, by the group of Fries in San Diego, reporting that mannose corrects the hypoglycosylation in fibroblasts of patients with CDGS. This was, of course, an enormous good news, and patients were started on mannose around the world, but the joy was of short duration because mannose did not help at all in these patients, even after long treatment and even after intravenous treatment for a long time, except in one patient responded, a patient in Germany. And when I heard about this, I made immediately the hypothesis that this patient should have a defect in the first step. So in the preceding step of the PMM2, in the first step, because in that case, mannose can correct the pathway, because we have the enzymes to transform mannose into mannose 6-phosphate, while our body cannot transform mannose into mannose 1-phosphate, unfortunately. So we could confirm this, and we and others reported this in 98, 3 papers in the same year on this disorder with a hepatic intestinal presentation. So these patients have no neurological problems. It is still a rather rare disorder. We know about 40 patients, reported patients. But anyhow, this is the first treatable, rather well treatable CDG. Then Gert Matthijs organized the first international workshop on CDG in 99. And then we renamed CDGS in Congenital Disorders of Glycosylation, because this is a more correct name. The previous name, you could not know if there was a defect in the anabolism or in the catabolism, while this points clearly to a defect in the synthesis, in the anabolism. But 10 years later, we had again to change names because the number was becoming so large that our nomenclature became a little bit, I would say, meaningless. CDG 1A, 1B, 1C, etc. We came at the end of the alphabet, also for the T, that was completely meaningless. And therefore, we decided to give it the name of the gene, followed by CDG, so that we knew that it was under the umbrella of CDG. And then we also made a very simple classification. In protein glycosylation, by far the largest number, N or O glycosylation. And then the lipid glycosylation defect are still a very small group, but I think it is potentially a very large group because we have an enormous amount of glycolipids in the brain, but they have still to be discovered. And then the GPI anchors have been added later on. Now, what has CDG learned, me and us? First of all, that you have to give attention to small changes, because small changes can hide very important things. It is a little bit like the stars in the night sky. A star is just a point, but it hides a universe. So you have to give attention to small things because this can be very important. Before CDG, the glycoproteins, at least the carbohydrate portion, it was not well known what that meant, what was the meaning of that or the function of that. It was more considered like a decoration. But now we know that it is really essential for life. And this is extremely well illustrated by CDG. The absence of a glycan or an abnormal glycan has really dramatic consequences for the patient. So the importance of glycosylation is here very well illustrated, I think. When you consider all the CDGs together, you have nearly seen all the symptoms or syndromes in medicine, or many of them at least, except maybe for a real cancer CDG. We do not really know a cancer CDG or a malignant cancer CDG, although there are in all cancers many glycosylation abnormalities. But I think that is a question of time, probably. Then some CDG have several codes, I would say. And I can illustrate this with the following. Multiple exostosis, a very frequent abnormality of the cartilage with benign tumors of the cartilage that can sometimes cause orthopedic problems and can exceptionally also become malignant. This is a dominant disease transmitted in all generations. And then Farran and 15 presented this family, this Mennonite family, with macrocephaly, scoliosis and epilepsy, and mutations in X2. So this was really very surprising, even more so because this was recessive. Exostoses are dominant. So this was a recessive syndrome with a completely different phenotype. Very surprising. Another surprise was this mutation of PMN2 causing polykidney kidney disease and hyperinsulinism. This was reported by Cabezas with large cysts in the kidneys, glomerular of origin, so glomerular cysts. And they found it in 11 families. Also in half of them liver cysts and real hyperinsulinism with hypoglycemia. And what other surprise, a normal serum transferrin isoelectric focusing. So this was very different from the classical PMM2 CDG, where you have normally logical involvement, where you have some dysmorphism, and where you can have hyperinsulinism, so increase of insulin, but without hypoglycemia. That is well known. You have several hormones that are increased, also growth hormone, but without consequences, let me say, because probably of a receptor defect. And the kidney cysts are also present in PMM2, but they are small and they are of tubular origin. And there are no liver cysts in this classical PMM2 CDG. And then, of course, we learned that parent associations can make a difference. And there, Vanessa has given us the example. She had a dream. Why did she have a dream? She explained us in the introduction. She has a sister with CDG, and she and her parents wanted to do everything possible to give her a life with the highest possible quality of life. And therefore she founded this parent association, and to boost awareness and research on CDG. And she has really made, I would say, really made a difference. She has given her energy, her time, and her skills to make this a model. Really a model. And now we have a model of how to involve parents, patients into our research. And is that not a dream team? Patients and all these professionals with a multidisciplinary approach. That's what these patients need, really. But there are also pitfalls. I mentioned just two of them. That is that the transferrin marker can normalize with age. So when you confront them at a little later age, you can miss the diagnosis, of course. And then another thing is, I think this is also important, that CDG is so heterogeneous. Most metabolic diseases are homogeneous. You have, for example, mucoviscidosis, and you have phenylketonuria that has, in fact, a very uniform expression. But here you have so many expressions, and that makes it difficult, I think, for parents and for patients to cope with it. And this is a problem for patient-parent associations. Anyhow, I think that 40 years of CDG research has given interesting fruits. Discovery of a large number of CDG reported in more than 1,000 papers. More important, of course, a large number of patients received a diagnosis, and even when there is no therapy, it is very important to have a diagnosis, to give them counseling, and to give them some mental rest. And I think it is anyhow important. Unfortunately, we have only 2 well-treatable CDGs. That is the MPI CDG that I mentioned. And then this recently discovered CDG, a defect in the pyrimidine synthesis that is important for making UDP sugars that are involved in the glycosylation in the Golgi apparatus. And here, uridine is really very efficient. A number of patient-parent associations have been established, and I think that there's also really a network of science and love around the world regarding CDG. But always think about CDG when you are confronted to a patient with an unknown syndrome. And I think there are many hundred CDGs still in the waiting room to be discovered. That is, there's no doubt about that. So this was really a story about great expectations, but also great frustrations, but also, I would say, coming back to the siblings, who are now 42 years, a happy family, and I think their optimism gives us hope that one day there will be a cure for this disease. This is the metabolic team in Leuven. Eva Mohava left us. She is trying to find out, I think, if Trump might have a new CDG maybe with narcissism as main presentation. And then we have, of course, our, I would say, collaboration over the years with the Gert Matthijs lab, and then Emile van Schaftingen and François Fourquier, and then the lab of Garozzo, Storiale, but also collaborations with our Portuguese friends and many others, of course, in the world. And finally, not forgetting Vanessa and Paula and this fantastic Portuguese-Spanish Patient and Parents Association that has really made a difference. And finally, I am grateful for your kind attention, and I wish you happiness and peace in the next year. Thank you.
For this wonderful talk, very inspiring. Are there any questions? I have a question. So the CDG are mainly or are recessive, right? But there are some cases where it is dominant, right?
Yes, and also X-linked also. Huh?
Yeah, but even in this case there are exceptions, right?
Yeah.
So you gave us the example.
Yes, dominant that becomes recessive. Yes. Okay. Yeah, that's right. But a few are X-linked. Yeah. But the large majority are recessive, of course. Yeah, that's right.
So there is always this is not a complete progression of the activity. Okay, there is always activity of the enzymes.
There's always some rest activity. Yes. PMN2, for example, there needs to be rest activity. There's a null, I would say, a mutation that gives a null result is not compatible with life. Mm-hmm. It's not compatible with life.
Maybe someone got more courage for questions?
Only a comment. As far as I understand at that moment, each patient is a case, or you need to have a specific treatment for each of the patients with CDG.
Yes,
so there is...
In fact, every disease is different. And you have to devise for each CDG another treatment, yes. That's right, okay. Absolutely.
And there is, at the moment in Europe, any kind of program specifically directed to this type of research, or for that?
Yeah, there are several groups in the world active on treatment. For example, in San Diego, the group of Freeze. There are also a few companies who are also involved in trying to identify treatment. Yes.
Yeah, but for example, as far as I know, in terms of Europe, for example, for neurodegenerative diseases, there is that joint programming that is trying to, I don't know, to join all the efforts from each of the countries and try to support with more money. But I don't think there is, because some rare diseases are represented there, but I don't think CDG has that kind of program, or I don't know. Yeah, also.
There were 2 elements, right?
Yeah, yeah, but this is still in another, okay, in another. Mm-hmm.
Yeah.
Which found 2 compounds, and they are now, they won now a project to decide the toxicity of these 2 pharmacological chaperones through a company called Enzonics, which does systems biology in Barcelona, and which are as well our collaborators in our network, because we want, as you said, we want to apply this, our next goal is to apply together with the European Reference Network on metabolic diseases for the next European call on rare diseases, focus on this.
So we are involved on
Yeah.
the metaverse, which is the...
No, I was thinking also trying to push a little bit to have a program, real specific program in that field. Okay, it's up to you also, because you are the one pushing.
We need to have someone fully working for that. We cannot burden the team with so many different tasks, because they are accountables, they are patient liaison, they are communicating with the families, they are fighting to get ethical committees approvals, which is a nightmare. So that's the issue. But I know you, and I want to speak with you.
But Vanessa has written also a very nice paper on the actual state of therapies. That is very complete. You should read this.
I have a question. I mean, then we would talk, but I was wondering about the hormones' role on these type of patients, because I mean, these patients grow, right? I mean, thank God. So I was wondering what kind of effects the hormones will have in them, even though I know that it's going to be pretty broad, the effects due to the heterogeneity of the disease. But, you know, if is there any type of information already available, or it's something that, you know, needs to be explored?
But they have hormone abnormalities. When we look at the PMM2 CDG, there are hormone abnormalities. There are insulin increases, there are growth hormone increases. Prolactin can be at birth extremely high, so that you would think about the prolactinoma, a tumor producing prolactin. But these, of course, secondary abnormalities, and they have no, I would say, no effect. It is not because they have a high insulin that they have hypoglycemia, because there are also problems at the level of the membrane, maybe particularly the receptors, so that the effect is blocked because of receptor defect. And that is a problem, of course, for the functioning of the hormones.
Hi. So, as I understand, this is a genetic disease, yes, and it is rather well identified which genes are in the origin of the disease or the many forms of the disease. So I cannot stop wondering whether this would be such a disease that would be, as it can be identified, you said it's recessive disease, so it is identifiable from the parents. So if we would go to genetic treatments, would it be possible to prevent the disease by genetic therapy? I mean,
by genetic therapy.
I do not want to go into more, let's say, unethical developments of the, or at least from some points of view. But so my point is, we could, if we wanted, prevent this disease in the future, if we would forget ethical issues. We know which genes cause the disease. We know we can correct those genes. Therefore, we could.
Yes, you can prevent it when you know already this. When you know already one child with a disease in the family, of course, but not the others, of course, because you never arrive.
If you know a priori that there is a possibility for it.
Yeah, then of course, but not you. So you can never prevent completely because every new child, it's a surprise. You will never know. You only know it when there is already a child in the family.
Gene therapy definitely for the most common rare diseases like Duchenne muscular dystrophy and many others is because they are very powerful economically and they are efforts in that sense. And also for lysosomal diseases.
Sorry. Sorry, I have because normally when you have for gene therapy, for CDG, the problem is that you have to tackle a lot of different systems because it's a multisystemic disease. Duchenne muscular dystrophy, it's a muscular dystrophy. The first papers, they inject it, directly in the muscle, so it's very easy to administrate. In lysosomal diseases, they take advantage of the change in pH to promote the liberation of the, because within the liposome or whatever they use to administer the gene therapy. With CDGs, you had to, devise a mechanism to reach all the different organs more or less at the same time, and that is the biggest difficulty that we have.
So nowadays, what are the main therapies that are generally used for treatment of CDG?
Well, Professor Yakin.
Yes, we have the manuals for the MPI. And there are also trials now on galactose because there are several CDGs that respond more or less to galactose, at least biochemically. Clinically also a little bit, but galactose seems to cause some aspecific defect on the genome. And there are several, 4 or 5 CDGs that respond to some degree to galactose. And then you have uridine for the CAD disorder. And there is also sialic acid for the GNE. GNE, there's another CDG that is mainly muscular CDG. So there are, but of course these are not really, will not be the final therapies. Finally, we have to devise some gene therapy, but I think we are still far from that,
So thank you so much, Professor.
Thank you.
So let's applaud you.